Every year, hundreds of thousands of people suffer severe spinal cord injuries. The resulting physical damage is almost always permanent. When the central nervous system experiences sudden, severe physical trauma, the body triggers an immediate localized panic response. Specialized cells called astrocytes rush into the injury zone. They interlock to form a dense, tangled barrier known as a glial scar. This biological reaction is called reactive astrogliosis. This scar serves a strict evolutionary purpose. It seals the wound and stops fatal infections from spreading to the brain. But the defense mechanism comes at a high cost. The scar creates a dense physical and chemical roadblock that severed neurons cannot grow past. Mouse biology and human biology differ at the cellular level, creating a massive barrier known in medicine as the translation gap. Researchers face an unsolvable ethical dilemma. To truly know if a drug works, you must test it on an injured human spinal cord, and scientists cannot intentionally paralyze a healthy human being just to test an experimental therapy. Until researchers can find a way to safely test these therapies on actual human tissue, bypassing the limitations of animal models, a viable cure for human paralysis remains out of reach. Researchers at Northwestern University have found a way around this ethical roadblock. They successfully grew a miniature human spinal cord entirely outside of the body. Look at this 3D rendering of an organoid, a 3mm tissue mass grown entirely from human stem cells. This 24-week timeline shows it generating real neurons, astrocytes, and immune cells. Unlike flat petri dish cells, this dense, three-dimensional structure perfectly mimics an actual human spine. Science now has a highly accurate human biological sandbox. Researchers can isolate the exact cellular complexity of a spinal cord without putting a single patient at risk. To test a cure for paralysis, researchers must first take these perfectly healthy human organoids and severely injure them. These diagrams show two trauma methods, a scalpel hemisection and a crushing impactor. Fluorescent scans reveal red patches of instant cell death. Weeks later, bright magenta cross-sections show massive astrocyte buildup. The organoid formed the exact glial scar that paralyzes human patients. By breaking the model and observing this identical scarring response, the researchers proved they had an accurate proxy for the human spine. The stage was finally set to test a cure. To defeat the glial scar, the Northwestern team deployed an experimental therapy, a synthetic nanomaterial composed of molecules called peptide amphiphiles. The therapy is injected as a simple liquid, but the moment it makes contact with biological tissue, it instantly self-assembles into a solid gel. At the microscopic level, this gel is constructed of millions of tiny, bioactive nanofibers. Their defining feature is supramolecular motion. The molecules are not static. They constantly dance, shift, and swap positions within the fiber while maintaining its overall cylindrical shape. This continuous molecular motion allows the material to physically reach out, locate, and optimally bind with cellular receptors. Once connected, they send powerful healing signals directly to the damaged tissue. This sequence shows the climactic experiment. Researchers applied a dynamic gel, IKAVAV-PA2, into the injured organoids. In these histology scans, the untreated injury displays bright magenta scarring. On the right, the treated injury is clear. The molecules prevented the dense glial scar. Look at this fluorescent scan. Without a physical scar blocking the path, the severed neurons responded immediately. We see a massive explosion of green neurite outgrowth as new axons rapidly extend straight through the injury site, penetrating deep into the blue gel. Scanning electron microscope images provide the physical proof. Thick, newly grown biological axons directly tangle and interface with the synthetic web-like therapy. These neural extensions naturally self-aligned along the nanofibers, establishing the physical foundation needed to rebuild the neural network. By validating this nanomaterial within an accurate lab-grown human model, scientists have finally built a bridge over the translation gap. A realistic, viable treatment for permanent paralysis has moved out of the realm of mouse biology and closer to actual human trials than ever before.